Alright, hey guys, welcome back to another video. And in this video, I am going to go over IB Biology 1, Cell Practice number 3. Now, Mr. Varseller said that in a couple of classes, not next class, not the next two classes, there will be a test. That's why he's been in this string of reviews. There's been multiple reviews now. I think there's been two. And he just wants us to be prepared for the next test because, I'm being honest, it's going to be quite difficult. Just looking at the questions on this practice, they are quite difficult. So, without further ado, let's start. Question number 1 states, what is essential for diffusion? Now, just like in the ATT and a lot of other tests that you're going to take, the process of elimination is really helpful for you. If you don't know what the answer is, that happens to me a lot of the time. Sometimes I just forget. I just don't know what the answer is. I just look at the other answers, and then I know, oh, that answer doesn't make sense, or oh, I know that isn't correct. And that's what happens with number 1 as well. Then again, some people might know off the bat what the right answer is. That's good. Good for you. You studied. But a lot of us, including me, I'm not perfect, they don't know the answer right off the bat. So, what I do, I look at, let's say, the product of the entire... What is the question trying to ask? What's essential for diffusion? Basically, what can occur so that diffusion can take place. Now, diffusion, if some people don't know, is any particle, doesn't have to be in a cell, it can be just a gas, disperses out from a concentration of low to high. And this can happen in the air. Let's say, even light, for example. So let's take this flashlight, for instance. So this flashlight right here. And there can be diffusion of light. Let's say this light is going from something super high concentrated to something super low concentrated, like out here. It's basically the same thing that's happening, but on a cellular level. So anything, any particle, any substance that's going from a state of high concentration or really bunched together to low concentration or dispersed out. And if you take that definition into account while you're looking at the answers, you can pretty easily figure it out. What is essential for diffusion? Do you need a protein? No, you don't need a protein. Do you need a source of energy for diffusion? You most likely don't. Because most of the time, when a protein, you need energy, and that's C as well. Sometimes you don't need energy with the source of protein, let's say for a channel protein. But you don't need energy or a protein for diffusion. You just need, let's look at B, a permeable membrane. Think about another analogy. Like my flashlight example, that was diffusion. Did I need a membrane? No, not really. Or something else. Let's say a bottle of perfume. If you open up that bottle of perfume and it disperses diffusion throughout the entire room, you don't need a membrane for that as well. A concentration gradient. Now that's kind of weird. If you don't know what that means, just break it down. So a concentration gradient is something like a difference, so like from this side to another. And then concentration, I think a lot of people know what it is. Like this is a high concentration over here, and then over here would just be like a low concentration or something. Just a couple dots. That kind of makes sense. From going to high to low, it's a concentration gradient. And that would be A. So essential for diffusion, you need a concentration gradient. Next, which of the following is a characteristics of organelles? Alright, so organelles, you know what an organelle is. That's just, let's say this is a cell. So inside of a cell you have organelles. I don't know, that thing, mitochondria. The nucleus, that's also an organelle. So let's draw that right quick. That was horrible, but you get the idea. So organelle is anything that is inside of a membrane, or a cell membrane, that does some sort of function. That in mind, let's look at our answers. They're found only in eukaryotic cells. Okay, if you think about it, eukaryotic cells, what's that? Okay, so we're made of eukaryotic cells, so like animal cells, or well, this is a plant cell. So like plant cells, animal cells, those are all eukaryotic cells. They're characterized by having a membrane and a cell wall. Well, this one doesn't have a cell wall, but nucleus. Mostly a nucleus and a membrane-bound DNA. That's basically what a eukaryote stands for. Let's keep that in mind because eukaryotes definitely have organelles, so that's correct. They're only found in prokaryotic cells. That doesn't make sense. Okay, so prokaryotic cells, what's that? That's like a bacteria, right? So, blah, blah, we got a bacteria there. Sad face. Bacteria don't have organelles, remember? They have free-floating DNA. They have ribosomes. Ribosomes aren't organelles. But they don't have organelles. So this doesn't make sense. Subcellular structures, you really don't know what that means because look, A. We passed A like it was nothing, but this is correct. There can only be one correct answer. So it has to be A. But let's just look at the other answers. You would just cross these out anyway, but let's go through them just in case you didn't know or you did A last. So see, there are subcellular structures. There are structures that are inside of cells. Well, there can be organelles outside of the cell. They won't live. But have you ever heard of mitochondria? Mitochondria were outside of the cell at one point. They were its own bacteria basically before our cells took them in. So it can't be C. D, they're all membrane bound. There are some non-membrane bound organelle. You could argue that cytoplasm is not membrane bound. So it can't be D, can't be C, can't be B. It's A because that's true. They're only found in eukaryotic cells. A. Alright, so number 3 is kind of tricky because Mr. Varesteller hasn't really talked about it. It's a diagram below shows a channel protein. Which of the parts of the surface of the protein would be composed of polar amino acids? Alright, that's a lot. If you don't know what any of that means, it's fine. We can break it down. Alright, let's see. What is it trying to ask? The diagram below. Okay, so this thing. It shows a channel protein. Now, that kind of makes sense. Channel protein, probably something goes through here, right? Maybe, I don't know, some sort of molecule. Let's say water. It doesn't have to be water, but let's say it's water. Which parts of the surface of the protein would be composed of polar amino acids? Okay, so which parts of this entire thing is composed of polar amino acids? I'm just shortening it to amino acids. Now, if you don't know what amino acid is, it's basically like a building block of a protein. So proteins are just a chain of amino acids like this. And that's a protein. But, I don't know. That's debatable if you need to know that in this context. But this is a protein and there are amino acids in it. Just think of it as like parts of the thing. Now, polar. What does polar mean again? So there are two types of molecules. There are polar, like this is, and then there are nonpolar. Basically the opposite. That's hence the non part. Now, polar means that it is charged. So let's take water for instance. Water has a slightly negative charge on this side and a slightly positive charge on this side because the electrons from this hydrogen more closely are attracted to the oxygen part. So it's charged. So water, always remember, this is key. Water is always polar. And if you imagine in a cell, there's water on this side, there's water on this side, and in between here there's water. So, you remember when we learned about phospholipid proteins? And how phospholipids are composed of the hydrophobic, this part, hydrophobic tail, and the hydrophilic head? Hydrophilic means water loving. It likes water. That's why it's on the outside, because there's water everywhere, so it wants to be close to the water. That's basically the same thing here. You want whatever's touching the water to like the water, which makes sense. So this part would want to like water, this part, this part, this part, this part, this part, because it's going to be touching water. And if it likes water, that means it's polar because water itself is polar. So anything that's touching water has to be polar. Just like these heads. They're touching water because they're polar. So if we look at it, 1 is touching water. 2 is also touching water. 4 and 3 is not because it's on the inside where there is no water. So 1 and 2 has to be polar, which is A. If I can circle it, there you go. A. Look. A. That's the grade that you'll be getting when you. It's just. Alright. So number 4. Distinguish between diffusion and osmosis. This is a question that comes up regularly, I would say. A lot of people ask me, oh, diffusion and osmosis, they're the same thing. Well, to a point. Diffusion, as I said earlier, is a movement of something, anything, from a concentration of high, to low. So let's say these particles again. So instead of it being like nothing on this side and then particles on this side, it'll just be some particles on this side, some particles on this side. More evenly distributed. Now osmosis, that's where it's different. Osmosis deals with water. So it's the movement of water from an area of low concentration, from high concentration to low concentration. So over here we have a beaker. And another thing about osmosis, it always happens through a semi-permeable membrane. Always. Diffusion, you didn't need that, just as question number 1. You don't need a semi-permeable membrane for diffusion. You need it for osmosis, though. And it's not just the movement of water from, like, high concentration of water. So let's say a lot of water here and a little water here. It's a concentration of the different particles in water. Let's see if that makes sense. So let's say you have some salt in here, and there's only 1 particle of salt there, but there are, say, many more particles of salt here. Now you can say this: the relationship between water to salt is really low because there's only 1 out of, let's say, 100 water molecules. Here it'll be 1 out of 99, because 99 water, right? 1 out of 99. Here would be 4 out of 96. So there's a higher concentration of these salt molecules per water. What osmosis does is they want them to be pretty much equal, like the concentration of this to this pretty much equal. So what osmosis is going to do, let's make the speaker bigger, it's going to move the water from this side where there's high to this side where there's a low amount of water. And this is going to fill up, and this is going to get a little less, because the water wants to go equalized. So the percentage of water to solute here should be about the same as here. So it'll be like more of 1 to 99 and like 1 to 99 here. Kind of the same. Now this kind of relates back to the question, because osmosis isn't just, oh, there's a lot of water over here, there's a little water over here, and it moves there. No, it moves from where there is a little bit of the solution to a lot. And it does this by multiple areas of a semi-permeable membrane, because this can't happen without a semi-permeable membrane. Otherwise, it'll just not work. Now, the only way that osmosis can occur, again, like I said, through semi-permeable membrane, just not like diffusion, because diffusion doesn't need anything. So, boiling it down, diffusion, anything, now has to be water, no semi-permeable membrane. Osmosis needs to be water. So, diffusion, anything, osmosis, water. Diffusion, no membrane, water, membrane. Alright? Makes sense. Okay, next. State the composition and function of a plant cell wall. It's kind of weird. It's A and B, but it doesn't have to do with each other. It's fine. So, a plant cell wall. So, basically asking us what is a cell wall made out of and what's the purpose of it. Now, if you think of a cell wall, where it occurs in bacteria. So, this boy. And then plants. This boy. Now, imagine a plant. A plant can go really, really tall. Like, the tallest plants are, like the California redwoods, are huge. And it does all of that without, like, a skeleton. Like, we have skeletons that stand ourselves up. But plants don't have skeletons. Plants have cell walls. So that's one aspect of it. That's the function. It's for support. And along with that, have you ever heard of, like, a plant cell immune system? Or, like, you know how we get sick and then we get better? A plant gets sick as well. But there's nothing really, like in our cells, it's all squishy, right? Like squish, 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 squish. But if you ever, like, touch a plant or something, it's, like, hard, like wood. It's pretty hard. And that's also for protection against, like, bacteria. And it's like a physical barrier between stuff. So it's also to protect. There you go. And how they do this, why cell walls are so hard, is part of the composition. It's made out of cellulose. And it's basically like, it's fiber to us, but it's a sort of sugar, like glucose, cellulose, fructose. It's a sugar, but it's indigestible to us. That's why, I don't know if you ever ate a salad. Okay, you've probably eaten a salad before, but it doesn't taste too good. It doesn't taste sweet, even though it's made out of cellulose, which is a sugar. It's because our bodies can't digest it. But it is still a sugar, technically. That's also the reason why it's so hard. It's made out of this sugar thing. Just like if you've seen table sugar, it's kind of like a rock. Part of the reason. So, composition, it's made out of cellulose. Function, it's for support and to protect it against either physical threats, let's say puncturing it, like just stab, or like bacterial threats, or life, I guess. Alright. And then, lastly, no, not lastly, number 5. The diagram below shows the structure of a cell. Let's zoom in this for a second. Usually when I have a diagram, I usually, like, label it before they even ask anything, because I know for a fact that they are going to ask about it. So, okay, these little dots. It's probably a right. Okay, just looking at it, there's no membranes, there's nothing, like, membrane bound. So you kind of know that this is a prokaryote. Right off the bat, because there are no membranes, there's no nucleus, you can see the DNA is free. Pretty textbook answer of a prokaryote. There's this thicker outing, it's probably a cell wall. Like we just talked about, it's thick, it's for support. There's another thinner inside of that, that's number 1. So wall number 1, that's probably like the cell membrane, so like cellular membrane. And it's like a normal cell membrane. Now this thing, I've actually never come across this, at least in his class. I know what this is from prior knowledge, but he hasn't taught us this. This is something that I actually had to look at a little closer and kind of remember a lot more. It's part of the cell membrane. So, part of how, remember when he was talking about surface area to volume ratios, and how that, that whole thing? Well, I actually learned that that little spool is called a mesosome, and it's spelled MESOSOME. And what it does, here, let me write that a little better, because we're going to need to know this. what that does is it increases the surface area of the cell membrane. mesosome, mesosome. And that increases the surface area of the bacterial cell wall. And remember that whole thing about like surface area to volume ratio, how it increased diffusion and all the rate of stuff. Yeah, that helps increase that. As you can see, like there's a lot of surface area to go there. Mesosome. This is just DNA. Another thing about it, I don't know if you can see it, but it's kind of circular. Like it's a circle. Like let's say you have a point here, and then you just go crazy like that, and then it meets up at the same point. So basically, if you bring it out, it's just one big circle. That's called circular DNA, and that's really accustomed to like prokaryotes. Only prokaryotes have that. Our DNA is just like, if you know, like double helix. Anything else we need to label? Oh yeah, these little dots are probably like ribosomes. Cool. So even before we labeled it, so we know what it is. Okay, let's look at question A. State the names of 1 and 3. Oh, look, we already did that. You go back, you look at 1, cellular membrane, 3, cell wall. There you go. So, cell membrane, cell wall. Cool. Alright, state the function of the structure label 3. Label 3, that's the cell wall. We just did that. Cell wall is for protection and for support. Alright, deduce which type of cell is shown in the diagram, giving reasons for your answer. So I already said that this was a prokaryote, and you can kind of see that from the DNA, from the cell wall, from there being no, like, any sort of organelle, any membrane-bound organelle. So we can write that as well. It is a prokaryote, and we know this because there is no nucleus. The DNA is naked or free, and there is no membrane bound organelles. There we go. And yeah, no nucleus, DNA is free, no membrane bound organelles. Good. Now you could also argue that another reason why you know that this is a prokaryotic cell, if we go back, if you look at it like super closely, you can kind of see that inside of here, like I said before, the DNA is circular if you look really closely. And that's also another reason why it's a prokaryotic cell, is the circular DNA. Let's try cDNA. But that's kind of sketchy. I wouldn't put that in it. I mean, if you need to. If that's, let's say, one more, I will put that on a test. Next, list 2 functional membrane proteins. Okay, membrane protein. So a membrane in a cell, that's a cell membrane. Proteins inside of the cell membrane? Okay, just remember that what that was. Proteins inside of a membrane. So 2 functions of it. If you remember that little saying that he gave us? The jet rat thing? The jet rat, that was way back. I made a better one, because that's kind of sketch. Like J, you know what J stands for? J, in his version, J stands for the connection between 2 cells. I mean, I don't know where you get the J from. E's kind of makes sense. Enzymes, T, transportation, R, recognition, A, anchorage, and then anchorage, that, like what? So, you can forget about J-Rat. I made a better one. So, it's C, R, A, T, E, R. Now, you can remember it as crater. That's fine. I think that's kind of lame, honestly. But the best way to remember it is cool raptors attack thick elephants regularly. Okay? Cool raptors attack thick, two C's. It's important. Elephants regularly. Cool elephants attack thick elephants regularly. Raptors. I said elephants. Now, each of these stands for C, connect. E, R would be recognition. A, attachment. T, transport. E, enzymes. R, receptors. Now, I'll write this all out. I kind of wrote it here. This is a different sheet of paper. If you want to pause and take a picture of this. But it's just, C is to connect two cells together. E is for enzymes. T is for transport. R is for recognition. A for attachment. R is also for receptors. This isn't in order, but you get the idea. So, just memorize that. We got connecting, receptors, attachment, transport, enzymes, and then recognition. Just remember, recognition, receptors, recognition, doesn't really matter as long as you got it down. So, two functions, to connect, let's say, and then attach. Well, those two same. Okay, the difference between connecting, so connecting two different cells, let's say you're taking this cell and then the cell right next to it, Minecraft like that, and then you connect it. That's connection. Attachment is something different. It's when you have, let's say, a cell, and then you're attaching it to its surroundings. That's the difference. So you can put connection and attachment, but if you're going to do that, then you would have to explain. I think it's easier to just put connection and then enzymes. That's not how you spell it. Spell it correctly. Alright. 6b. That is oxygen moves across the membrane by diffusion. This term define diffusion. Simple. We already know what it does. Let's just put it into words. So diffusion is the movement of molecules from a higher concentration to a lower concentration. There you go. Now, the way that potassium can move across the membrane by passive or active transport. Distinguish between active transport and facilitated diffusion of ions. That's quite a big question. So it's just saying potassium can move across the membrane by passive or active. So basically you're distinguishing between active and facilitated transport. Now, I made a huge array of notes a while back during class that displays a lot of the transport modes. You can see here, if I can get the entire page into view. It's got a lot. That's good enough. But yeah, so we have diffusion, osmosis, facilitated, and active transport. For this question, we're just focusing on facilitated and active transport and the differences between those two. So, if I can get that, there you go. Let's see. Potassium can move across the membrane by passive or active transport. Distinguish between, okay, so we're just distinguishing between them. Good. So let's see. Facilitated diffusion. It's kind of hard to see, but I'll say it. With the help of proteins, which channel the substance through with the concentration gradient high to low. So facilitated diffusion uses proteins to go from high to low. As you can see, high concentration over here, see proteins here, low concentration here, make them even. Now, active transport is where it gets crazy. Uses energy to go against the concentration gradient. So it's going from low to high. See how it's low here, it's high here. It's going that way. And it uses energy, uses ATP. See ATP. That's basically what this is trying to say. Active transport uses energy. Let's do it in blue. Active transport uses ATP, adenosine triphosphate. I spelled energy wrong, but you know what? You get the idea. It uses energy to go against the concentration gradient. Again, spelling, it's fine. So it's going from low to high. Facilitated diffusion, active transport, uses proteins and it goes with the concentration gradient. And it's going from high to low. Now this also uses proteins. I'm not saying that this doesn't use proteins. This also uses proteins. Alright. D. The hormone insulin leaves the cell by exocytosis. Describe the process of exocytosis. This is another thing he did not teach us that I'm going to have to teach to you. I'm going to spend quite a bit of time to this, so yo to all those people skipping to this point. And we are going to go over exocytosis. Let's make this in a big, bold thing. Now, the question just describe the process of exocytosis. So if you just want to go straight to the answer, be in the link description below. But he didn't teach this to us, so I think it's really valuable if you stay and understand what this is. Because I did a couple of research on this. So let's do this. Exocytosis. Now, exocytosis. This is the process of any substance inside of the cell. So let's write that down. Any substance inside of the cell to leave the cell by way of vesicles. Now, that's a lot of words. So let's break it down. Any substance or anything inside of the cell. So this is going to be inside. And that's with exocytosis. To leave the cell by vesicles. If you don't know what vesicles are, they're basically like a mini cell. It doesn't have like organelles and stuff, but it's just membrane bound, and it's inside of it. That's a vesicle. It's inside of the cell, and it has stuff inside of it, the substance. So let's draw this out. Let's say we have this cell here, it's minding its own business. It wants to exclude, just draw a quick nucleus here, and it wants to exclude some insulin. Let's say their insulin built up here, doesn't really want it, and it wants to exclude that. So it's going to undergo exocytosis. Now you can remember it's exiting because it's exit, leaving. Now let's zoom in here. So here we have the cell membrane, and here we have the insulin. So step one for this, so say step 1, is for the stuff inside of the cell to grow a vesicle. Let's have this I and then out, inside and then out, so we can keep it straight. So it to grow a vesicle inside of the cell. It's vesicle. Vesicles. And what that does is it puts the same stuff that's in our membrane proteins, so the phospholipids, all around the substance that wants to be removed. And then over here, say step 2, this vesicle joins with the cell wall, the cell membrane, and it links towards it. And what it does is, once it's linked, this is step 2, it kind of degrades the cell wall a little bit, so that the next step, step 3, you can kind of see right here, if you zoom in, that there's a little opening. And now it's not just going to go through this little hole, no. Actually the whole thing is going to grow, so it's just going to be like this. And then all the substance just leaves. Entire substance just leaves the cell. And then it's just going to be a little wrinkle there, and then just goes back to normal after. And then all the particles are out there. Step 4, and then this will be step 5. And that's the process of exocytosis. It's basically going from a substance inside of the cell, growing a vesicle, joining, making it part of the cell wall, and just excluding it. Now that's what happens in question number 6. So I displayed it. That's exocytosis. Be sure you know that, because he's going to go over it. Describe the process. You can say, you can draw. It's saying describe, so you can't draw. By way of vesicles, exiting the cell. Joining the cell wall. And exiting. That's a lot of exiting, but that's the whole point. Existing. Sure. Cool. So that was practice number 3. This is the simplified version of the huge practice that he gave us. So the thick packet. But I'm going to cover that in my next video. So stay tuned to watch that. Alright guys. Hope you learned some stuff. Bye. Okay guys, a quick overview of cell practice number 3. So for question number 1, what is essential for diffusion? A concentration gradient, so diffusion going from place to another. For number 2, which of the following is a characteristic of organelle? A, organelles are only found in eukaryotes, not in prokaryotes, nada. 3, it's also A, so water wants to go towards polar, so water is also polar, so they're going to be touching these polar ends. So these also have to be polar. So 1 and 2, those have to be polar because they'll be touching water. 4, distinguish between diffusion and osmosis. So diffusion deals with anything, non-charged particles. Osmosis deals with water. State the composition and function of a plant cell wall. Plant cell walls, they are composed of cellulose, and they're used to support and protect the cell. The diagram below shows the structure of a cell. So I annotated it before. We don't need to know that. Question number 1 states, what is 1 and 3? So if we look at 1, 1 is a cell membrane. 3 is a cell wall. State the function of a structure number 3. So basically, state the function of a cell wall. Cell wall is to protect and to support. Define which of the cells is shown in the diagram, giving reason for your answer. So what do you think this is? I think it's prokaryote, and the reason is because it does not have a nucleus, it does not have DNA, does not have any membrane-bound organelles, and its DNA is circular. Now, I like A6A. List 2 functions of membrane proteins. Remember, cool raptors attack thick elephants regularly. Cool meaning to connect, so connect to cells, are meaning receptors. A, attachment, attaching to a surface. T, transport, transporting materials. E, enzyme, so enzymes on the surface. R is for recognition, so cells can be recognized. So 2, you can pick any two of these to connect or to attach. For B, oxygen goes by way of diffusion. Diffusion is just the movement of molecules from higher concentration to lower concentration. Pretty simple. Okay, for C, potassium membrane by passive or active transport. So you just have to do the difference between passive and active. So active uses energy, uses enzymes, and it goes against, not enzymes, uses proteins, but it goes against the concentration gradient, and it goes from low to high. Facilitated diffusion uses proteins and goes with, so it goes from high to low, basically the opposite. Does not use energy. D, hormone insulin leads the cell by exocytosis. Describe the process of exocytosis. Exocytosis I labeled in the quick sketch I made. Oh, I made a different sketch here. Actually, if you want to see this sketch, which I think is better, you want to quickly hop over to cell practice number 4 and then go into the description and look at the times, and I'll explain this. But for what you guys saw and the one that we made quickly, exocytosis is the process of material inside of the cell latching onto the cell membrane and releasing. And putting that into words by way of vesicles. There's no T. It's V-E-S-I-C-L-E-S. There is no T. Existing in the cell, joining the cell wall, and exiting. Alright guys, that is a super quick overview of it. If you want to go back and look at how I did or understood different questions, just go back in the description and they'll all be there. See you guys in the next video. Hope you get an advance on the test.